The brain is one of the most mysterious topics, something that we as humans still don't understand all too well. Yet here we are, doing our best to replicate, reverse engineer something that we do not completely understand. Forming a deeper, more knowledgeable understanding of the brain could lead to better computing capabilities, the curing of brain disorders, reconnecting neural signals back to paralyzed limbs, and many more novel scientific breakthroughs. It is a grand endeavor indeed, and in order to accomplish it, each part of the brain must be studied diligently. For our part, my colleagues and I have amassed quantitative data on neuron activity at the cellular level. The electrical responses of neurons happen to be the simplest activity in the brain, making it a good place to start. By expressing this data in a graphical manner, we intend to gain further understanding of neurons and thereby the brain in general, bringing us another step closer to accomplishing the grand challenge. The goal of this data set is to show the modeling of somatic and dendritic spike plasticity of a single neuron. We can take this data, use its equations, and the study of the model, and translate it to a user-friendly format. We hope to show the simulations of the neuron models and display raw data that can easily be shown to the user by translating the scripts from the folders into the matrix laboratory. We want to be able to actually quantify and visualize how a single neuron works, and at the same time provide a framework that would allow for the comprehension of the broader neuron networks in the brain. The user know what the data is being used for and give them an option to view the simulation of the model and allow them to check certain regions they want to see by clicking on options that will also be provided through a menu. The user could also be given another option to view plotted data on the graph. Our overall ambition is to provide a simulation showing a glimpse of the live dendritic spikes that are constantly occurring in the
In
brain.
order to follow our code, we use a file called Bronco 2010 and Acura 2008, which are SWC files. The files originate in SWC format and is a text file with a header and contains number characters. It has a series of three-dimensional points containing an index, radius, type, and connectivity information. The first column is sample number, second column is the structure identifier, third, fourth, and fifth columns are X, Y, Z coordinates respectively. Sixth column is the radius, and seventh is the parent sampler of the sample number. First, we convert the file to an XLS file so it can be read and loaded onto MATLAB. And the variables are names for parts of the neurons. And our code here checks each row for types of neuron parts, and if it is a parent, it is a child variable connected to the parent part. We use a for loop in order to check each row of the matrix. We use length without warning about sizing because the depth SWC format dimension should always be 7 columns. The if statement checks which the second column is the structure identifiers and determines which part of the neuron the data is a part of and assigns it into a vector with that variable name. It then finds the XYZ coordinates for the variable names and stores them in a vector. And this code looks through the matrix and finds any place that the radius is 0 and moves it in the same index.
So each node, like the soma, the axon, and all of those things, each node has exactly one parent, one pathway that leads to it, and it may or may not have one or multiple children, or maybe no children at all. So for this area, we just identify which parent it belongs to identified by the sample number and the amount of children it may or may not have. So from 1 to the sample which from the previous loop, it should have the amount of rows that we go through. Fill it up with empty arrays for the child, so that hopefully we would help the computing time.
The current plan is.
And now, if the child cell array has been all filled out, we would just go through, find the corresponding parent, then fill it up with the fill each cell array with the corresponding children. So in this case, the index would actually refer to the specific sample, such as if it was 1, then it would technically be the soma, if it was 2, it would be the axon, then so on and so forth. So once we have the parents and the children, then we can go to the next part where we actually graph the pathways between between all of the nodes. So this view 3 and hold on is really just to make the graph 3D. So initially, this entire loop here was really just to identify which samples had multiple branches, which samples had ending branches, and if there was just one single branch, then that would have just left it empty. So we're just trying to graph so see, it depends. So if it had one parent, so we know that which parent it goes to, and then but the idea is that we want to know the nodes, how many children comes out from it. So it will run differently depending on whether it had only one children. If it had only one child, then it would just be a straight line that goes out. Or it would be this little dot with a flower kind of thing that would just spark out from it, or it would just be a stick that ended. So with this, I'm not sure if I can explain this very clearly. It took me an entire Saturday afternoon that I had to spend working on this instead of, I guess, sleeping. So for the A, that was finding the corresponding parent, for the so-called B, which I wasn't really too creative about naming. You would find the corresponding children, and depending on whether the arrays of children had more than one element, no elements, or I guess you could say there's just one element, then you would just graph from that node to each of the children in sparks. And well, explaining it is actually a bad move. I would actually run it. Let's run. So as you can see here, the multiple branches are right here, the ending branches, and so on. So let me close this one. So as you can see, the paths here they depend a bit on how the A's and the B's run. These are the X coordinates, the Y coordinates, and the Z coordinates that connect all of it to it. So as you can see here, you get a big flower shape, or I guess a flower shape is yeah. And then, where did it go? Yes, and then down here are just the nodes, which are represented by spheres for the radius with radiuses. Yeah. So our program is a really simple imaging program. That's all it really is.
Although our project it works, it's limited by a number of factors. So our dataset can't be too large, or at least not like ridiculously large. The speed at which our code can process is limited by the size of the dataset. The Accurate 2008 dataset, for example, is very, very large and is very stressing on the system processing. And so a larger dataset of, let's say, an entire ganglion might be too much for the system to handle. A dataset also cannot comprise of too many spaced out data points. At some point, the dataset will be essentially unreadable at a certain scale. The second figure, for example, would lack indistinguishable points, and the user would not be able to differentiate between different nodes and other points. So we lack the ability to view such a dataset flexibly. The code also has to plot two figures. We couldn't figure out how to best put them together. We also don't know whether or not the large sphere on figure 2 is really there or not. What you do know is that we have to plot a unit circle onto the figure, or else the data will not show. So there is a rogue data point there somewhere. But though our code is limited, it can still be utilized to map out small portions of nerve bundles. Professional biomedical engineers or those in bioinformatics can use this system to revisualize sections of ganglion or nerve tissue to identify probable misconnections, or in general, identifying specific types or sections of neurons within a specific area. And future work on this code could
work to animate the electrical impulse along the neural paths in order to better visualize the function of the neural network. Other than that, you could also allow for other data sets to be built upon the prior data sets, which would basically allow for the interaction between bundles of neurons to be seen.